We want to know how the brain works during perception and behaviors. Zebrafish larvae can recognize a paramecium at 5 days post-fertilization. The fish finds the paramecium, approaches the paramecium, and catches it. We aimed to visualize the brain activity during perception of a paramecium. In order to make this possible, we developed tools and methods in the model vertebrate zebrafish. One important tool is GCamp. GCamp was invented by Dr. Nakai and Dr. Okura at Saitama University in 2001. GCamp contains M13, GFP, and calmodulin sequences. When calcium ion binds to the calmodulin sequence, conformational change occurs and GFP fluorescence increases. Because calcium influx occurs when a neuron is activated, we can use GCamp to detect neuronal activity. Further, in this study, we developed a new GCamp, which is more sensitive than the old GCamp. Another important technique is the GALFO-US system. GALFO binds to US and activates transcription of a gene placed downstream of US. In my lab, we are doing gene trap screens and generated more than 1,000 transgenic fish lines that express GALFO in different tissues. Each tank has a different line.
Animals, including humans and fish, have common features in the brain structure. One of such features is the visuotopic map. What it means is that neurons in the brain are spatially organized so that the visual world is mapped on a part of the brain. This is a well-established notion, but no one has ever demonstrated this in real time in a natural condition. In this study, we wanted to visualize this using our new GCamp in prey capture behavior. In fish, the optic tectum is the center for visual information processing. So first, we looked at our database and chose one GALFO line in which GALFO was expressed in the optic tectum. On the other hand, we generated UAS GCamp7A transgenic fish. By mating that GALFO fish with US GCamp7A fish, we expressed GCamp7A in the optic tectum of the larvae. The experiment is straightforward. We let the paramecium swim around the larva and do calcium imaging using fluorescence microscope. Here's what you see in the recording. Changes in fluorescence intensity is shown in pseudo-colors. Colors on the eyeballs are artifacts due to the image processing, caused by eye movements. Neuronal signals are observed in the optic tectum. We could also record from a free-swimming larva which captured a paramecium. We visually demonstrated that the paramecium was represented on the visuotopic map as predictable by anatomical studies. And activation in the anterior part of the optic tectum was correlated with the occurrence of the prey capture behavior. In this study, we imaged neuronal activity in the initial step of prey capture. Using this method that we have developed, now we are trying to visualize the neuronal activity that underlies cognitive process.